Proteomics is the large-scale study of proteins, their structures, functions, and interactions within a biological system. Proteins, being the functional molecules of the cell, are critical for understanding cellular processes, disease mechanisms, and biological pathways. Proteomics complements genomics by focusing on the end products of gene expression, giving a dynamic view of biological systems. Proteomics encompasses several specialized branches. Structural proteomics focuses on determining the three-dimensional structure of proteins and protein complexes. It involves techniques like X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy. Functional proteomics studies protein functions, interactions, and their roles in biological pathways. It explores post-translational modifications, such as phosphorylation and ubiquitination. Expression. Proteomics analyzes changes in protein expression levels under different conditions. It is often used to identify biomarkers for diseases or responses to treatments. Interaction proteomics investigates protein-protein, protein-DNA, and protein-lipid interactions, helping to understand signaling pathways and molecular mechanisms. Quantitative proteomics measures protein abundance in a high-throughput manner, utilizing methods like isotope labeling, such as SILAC and ITRAC, and label-free quantification. Clinical proteomics focuses on identifying proteins as biomarkers for diagnosis, prognosis, and therapy in medicine. Lastly, metaproteomics studies protein compositions in microbial communities, such as microbiomes. Bioinformatics tools are essential for analyzing the massive amount of data generated in proteomics. Common tools include protein sequence analysis tools like UniProt, XPacy, and FAM. For mass spectrometry data analysis, tools such as Mascot, MaxQuant, and X-Tandem are commonly used. Proteome Discoverer is another important tool in this category. For protein structure prediction, AlphaFold, Swiss Model, and ITASA are key tools. Protein interaction networks can be studied using tools like String, Cytoscape, and Intact. Post-translational modification analysis involves tools such as PTMscan, NetFOS, and ModPred. Quantitative proteomics tools include Skyline, Perseus, and OpenMS. Functional enrichment analysis can be performed using David Go enrichment tools and KEGG. Finally, metaproteomics analysis is facilitated by tools like Metaproteome Analyzer and MetaQuantome. Proteomics is vital for understanding cellular processes and provides insights into health and disease. Some key aspects of its importance include deciphering biological complexity. Proteomics reveals how proteins interact, function, and are regulated in cellular contexts. Another crucial point is dynamic insight. Unlike the genome, the proteome is dynamic and reflects real-time physiological states. When it comes to disease understanding, proteomics identifies disease-specific protein patterns and biomarkers, helping us understand the molecular mechanisms of diseases. And as a complement to genomics, proteomics explains phenomena that cannot be understood solely through genetic data, such as post-translational modifications. Proteomics has wide-ranging applications in various fields. In medical applications, it aids in biomarker discovery by identifying protein markers for diseases like cancer, Alzheimer's, and diabetes. For drug development, proteomics targets specific proteins in drug design and development. Precision medicine also benefits, tailoring therapies based on a patient's proteomic profile. In agricultural applications, proteomics improves crop resilience by identifying stress-responsive proteins and studies plant pathogen interactions for disease resistance. For environmental applications, it analyzes microbial proteins for bioremediation and pollution control, and studies the proteomes of organisms in changing ecosystems. Industrial applications include optimizing enzyme production for industrial processes and developing biocatalysts for pharmaceuticals, biofuels, and food industries. And in basic research, proteomics elucidates molecular pathways and cellular functions, advancing our understanding of evolutionary biology and protein evolution. By integrating advanced technologies like mass spectrometry, bioinformatics, and machine learning, proteomics bridges the gap between genomics and phenomics, driving innovation in science, medicine, and industry. Nature is ultimate.